Now that you learn how to use the microscope, the next step is how to prepare your very own wet mount slide. We're going to refer back to this podcast often because a lot of the activities that we do will relate to preparing your own wet mount slide. So the first step is to get a clean slide, and you're going to have a stack alongside your microscopes ready to go at the beginning of the lab. Just place a clean slide onto the work surface, and you're going to need to take a small sample and place it in the center of the slide, like so. We're going to do two samples in the first case here, our first lab activity with a microscope. First one is the cheek cell sample, and you're going to take either a Q-tip or a coffee stirrer. No sharing of samples here. You're going to do your own sample, and you're going to sweep back and forth just between your the base of your teeth and your cheeks to get a good sample. And then you're going to place it in the center like so. Also, you're going to take the smallest piece of Eladia leaf from the top, the young leaves, and I want you to take that and then put it also in the center here. So you're going to have two slides, one from the cheek cell and one from the Eladia. You're going to place that small sample in the center, and then afterwards you're going to place a drop of distilled water on top of your sample. Usually we use distilled water, but in this case you can use tap water, it's no big deal. So bloop, one drop. After you place that drop, you take a cover slip, which is alongside your slides, place it, lower the cover slip into place. As you hinge the glass downward, the drop is going to spread because of the capillary action towards the cover slip, and it's going to spread outward, and suspend the sample in between the slide and the cover slip. You want to minimize the amount of bubbles that you form. For optimal viewing, I want it to have the least amount of bubbles as possible. And you'll see as you are looking at it. So that's it. After you're done with that cover slip, you're ready to place it in your microscope for observations.